Good afternoon, everyone. My name is Alessandra Borgognone. I'm a postdoc in the Irsi Institute in Barcelona, and I'm going to present the results of our study regarding gut microbiome signatures linked to HIV-1 reservoir size and viremia control. One of the main clinical problems with HIV, vaccine and immunotherapies is the persistence of latently infected cells. So one way to try to eradicate HIV is by giving patients who are on antiretroviral treatment vaccine combined with latency reversing agents, which are expected to make latent cells produce viruses. In the BCNO2 clinical trial designed by Christian Brander and Bea Mote, assessing such vaccination strategy called kick and kill by combining MVA vaccine with romidepsin given as latency reversing agents. 3 out of 13 patients were able to spontaneously control HIV replication during 32 weeks of a monitored antiretroviral interruption. So in our sub-study, we wanted to investigate if the composition and functions in the gut microbiome in these 3 individuals able to control viral replication could predict the clinical trial outcome. To accomplish this, we used a multi-omics approach as outlined here in this workflow. In the preliminary results presented last year, we showed that despite the small number of patients who were viremic controllers were enriched in bacteroidales and depleted in clostridiales. Therefore, the bacteroidales-clostridiales ratio was significantly higher in this group of patients, and this observation was found before vaccination already at the baseline, and during the intervention, this association more or less was maintained. These results were also recapitulated at the species level analysis, where we found that viremic controllers were significantly depleted in clostridial species, generally described as butyrate-producing bacteria. Another interesting finding was that viremic controllers had lower microbial gene richness and diversity, as you can see here by this gray and pink box plot. And moreover, we almost were not able to detect any methanogenic archaea in viremic controllers compared to non-controllers. So differences were very high also at this level. So the signatures associated to the gut microbiome were pretty clear and were preceding the vaccine intervention. So we wanted to further focus on the baseline to try to identify further signature potentially supportive of this observation. Looking at the PBMC transcriptome, we found functional enrichment in neutrophil-mediated immunity, neutrophil activation, humoral immunity response in viremic controllers, in which previously we also found depletion in anti-inflammatory bacteria. Also, we found a number of plasma protein inflammation proteins increased in viremic controllers, possibly indicating that these subjects had higher level of innate immune activation and humoral immune response signatures. A very interesting finding for us that we are going to present it here for the first time, and also for the HIV-1 field in general, was that the bacteroidales clostridiales, baseline bacteroidales clostridiales, negatively and significantly correlated with the viral reservoir size, measured at both HIV-1 DNA and cell-associated HIV-1 RNA at the baseline, but also over the intervention, whereas the gene richness, which was found lower in viremic controllers, was showed an opposite trend. In addition, Adam Burgener's lab looked into the bacterial proteomics before vaccination, and they could cluster non-controllers and viremic controllers very clearly, and controllers were actually enriched in functions mainly related derived by Blautia and Romincoccus, suggesting that Clostridiales-derived protein will discriminate between viremic controllers and non-controllers. And finally, the integration analysis of all these factors showed that Bacteroidales and Clostridiales and their related proteins, as well as annotated transcripts related to immune activation, were able to discriminate between the two groups, as you can find here, visualized as purple and blue lines. And moreover, both cell-associated HIV-1 RNA and HIV-1 DNA negatively associated with transcripts related to neutrophil-mediated immunity and humoral response, as well as Bacteroidales species, as you can see here visualized, represented by these red lines, whereas antimicrobial and butyrate-producing, butyrate-producing species such as Bacterium cereum, Suprogranulum, and Dorea were positively associated with the viral reservoir size. So our interpretation is that although this is a very small study, what it was suggesting was that patients should have certain levels of immune activation in order to be able to control HIV replication spontaneously or after a vaccine intervention. And this may be in a certain way reflected to the gut microbiome. So we cannot definitely establish the causal relationship between the presence of such microbial patterns, the pro-inflammatory signatures, and the viral reservoir size, but our research suggests that these aspects can be somehow significantly related. So in the next step, we first want to validate these findings in a larger clinical trial, and this is ongoing in our unit. And if this observation are confirmed, then we could at least predict beforehand who could control HIV replication, and then maybe select more precisely which patient may better benefit of this treatment. To conclude, I want to acknowledge all the people that have been involved in this study, and thank you all for your attention.